Okay, I know it's late in the afternoon, but I saw snacks over there, so hopefully everybody had a chance to get a little bit of sugar. Really pleased, obviously, to be here again this year and to be talking to you about microgravity and how we can use that as an enabling technology. Before I start, I just want to point out who's on our committee and actually have them stand so that you can see Dr. Arun Sharma, who's here, and Dr. Mary Kearns-Jonker, who's in the back. Both have flown missions on station with us. Dr. Abba Zubair, who couldn't be with us from Mayo Clinic, has also flown research on station. Anthony is with us, Zorana from NASA, from part of our NASA team, and Kunal, is he still here? Yeah, perfect, from FIT. So our group is a great group that brings experience both from the NASA side as well as researchers who have done work on station and others who have deep space background and science background. We welcome anyone who would like to come and talk with us and be a part of this group. There are now several other researchers who are doing research on station as well. Certainly, we would love to have those in our group to help us to direct how we look at some of these activities. There we go. So we put very simple slides together because we felt like our group touches on so many different areas. You heard me say just a few minutes ago when we were talking about the musculoskeletal tissues, you know, obviously station presents an opportunity to study certain disease states in a way that is much more powerful than we can do here on Earth. But the conditions in microgravity and the changes that you see in gene expression, cell signaling, the way cells aggregate, the way they interact, matrix deposition, all of those pieces really are integral in terms of answering questions across the challenges that we face here. So we just took a look to sort of say within our committee so far, what are the questions that we could ask and answer through research on station that may really help to advance the field? And the first area that we came up with was sort of the big question, what makes a stem cell a stem cell? And what causes differentiation in stem cells? That is sort of a fundamental question of tissue engineering, but we thought that it would help both from a cell sourcing perspective for us, as well as growth factors that are involved in that process, even patterning and morphogenesis, really, how those tissues actually change over time. Interestingly, we've found with stem cells that they grow a little quicker on station and their differentiation is a bit delayed. So when you start to talk about therapeutic populations of cells, it's possible that microgravity could really lend a very nice hand in helping us to get those larger numbers. We heard a lot of you talking today about the fact that you need millions to hundreds of millions of cells. So if there's an opportunity to kind of accelerate that process in microgravity, that may help from a therapeutic perspective. The other area really is what happens with networks like vascularization. Obviously, we're part of the Innovations in Space Award, which is a follow-on to the Vascularization Challenge, but really that whole process of how cells deposit matrix, how tissues integrate into the body, and sort of what we can learn in microgravity are areas that we felt as a team we could add across the challenge topics and foundational and scalable challenge topics. How do cells organize? This is another big question about patterning and morphogenesis. I think cell mapping and integration, we've talked, everyone who stood up today and talked about their individual tissues talked a little bit about how they need to understand better what happens in that developmental process of the tissue and how it integrates into the body. So I think you're aware of the work that we've been doing on tissue chips. We see that as sort of the starting point, right? When you talk about tissues, you have to start somewhere. So first you start on chips, then you can make small pieces of tissue. I like to call them the biological Band-Aids, and then you get eventually to the whole organ. So we have been very fortunate to have a collaboration with NIH. We did a tissue chip solicitation where actually 5 different tissue types are actually selected to fly on station, and we just closed another solicitation with NIH for tissue chips 2.0. So we're expecting to see even more of a variety of tissues that will come in, but again, gives us a starting point to really understand small models, tissue chips, models of disease, models of different cell types that we can look at in microgravity where we may be able to get some directional information that will really help in the full tissue engineering efforts of tissues that are moving off chips and into pieces of tissue that can be transplanted and integrated. And then the last area, we looked a lot at how do we help some of the other enabling technologies. So, for example, Anthony's group actually created rotating wall vessel bioreactors for station and different pieces of hardware that allow us to do research in microgravity. Some of that engineering could lend itself very nicely to the development of bioreactors here on the ground. Same thing for bioprinting. We had a big conversation this morning about 3D bioprinting and how we can really move the field of tissue engineering forward. Same is true for the work that's happening on station. And what we find out in that microgravity environment, I think Gene didn't mention it this morning, but I've heard him in some of his talks before talk about the fact that you really, on the ground, it's still somewhat 2D printing. When you get into a microgravity environment where you really can place polymer or matrix proteins where you want them, it gets to be a little bit interesting in terms of the geometries that you can create. Biodegradable scaffolds, right? Areas that we all need to start in any tissue that we're talking about. A lot of discussion today that I heard was, should it be fibrin? Should it be collagens? Or should it be synthetic materials? And how much of it should be natural? All of those pieces potentially could be very helpful for us to understand tissue specifically, but across the board. So we're very pleased to have put a collaboration together with NSF. Michelle is here, and we just closed that solicitation, and we're very anxious to see the types of research that people are proposing for tissue engineering. But it does span several categories, and we hope that we're going to be looking at things like scaffolds and systems that will allow us to move the field forward. In terms of the specific tissues, one of the reasons that I wanted to make sure that everyone is aware of Arun and Mary today, so they've done work with cardiovascular tissue. We have work coming up on tissue chips that will cover the kidney areas. We don't have anyone with liver yet, but we do have a lot of muscle and bone research that's been done on station as well. So some of those areas that are tissue specific, we already have some published literature or research that's been done there that could really be helpful. So again, even within your groups, come and talk to us if we haven't been able to connect with you at this point. And if we can share information for what we're finding in that environment where we do see different conditions that allow us to see things from a very different perspective, happy to share that and add it here. What's next? Hopefully a lot more research on station, and hopefully really getting to that goal of ending the organ shortage. It's fantastic to see the collective effort, and I look forward to seeing you all next year again.